Somewhere in the Altai Mountains of Siberia, there is a cave that carries only the name of a forgotten hermit. And inside that cave, buried in layers of earth older than any written record, scientists found evidence that 3 different human lineages once passed through the same place. Not in the same year, not around the same fire, but close enough, in geological time, that the word together is not entirely wrong. For most of history, we told ourselves a simple story. One kind of human mind spread across the earth and inherited the world alone. The cave suggested something else. That when Homo sapiens entered Asia, the continent was already inhabited. Neanderthals were here. Denisovan-related populations were here. Other human lineages, some known and some perhaps still undiscovered, were already moving through these landscapes long before our ancestors arrived. And in at least some cases, the boundary between them and us did not hold. Tonight, we go back to the cave where those traces were found, to the continent that was, for thousands of years, shared territory, and to the possibility that part of that ancient world is still present in you now, in the silent biology of your cells. We were not alone when we arrived in Asia. We may never have been as alone as we imagined. Settle in. Let your breathing slow. The past is patient. It can wait a little longer. Chapter 1. The Cave with No Name. There is a specific kind of silence that belongs to places where very old things have been found. Not the silence of emptiness. Not the silence of a room where nothing has ever happened. The other kind. The silence of a place where so much has happened, across so much time, that the ordinary sounds of the present feel thin and provisional by comparison. The silence of a place that has been waiting longer than you can properly imagine. Denisova Cave sits in the Altai Mountains of southern Siberia, near the border where Russia meets Mongolia and China and Kazakhstan, in a knot of high terrain that has always been difficult to reach and easy to overlook. The mountains here are old in the way that very few landscapes still are. The rivers run cold and clear. The forests are dense with pine and cedar. The winters are long and severe in ways that most people who have not experienced them cannot fully picture. The cave itself opens into a limestone cliff above the Anui River. It is not a dramatic opening, not the kind of entrance that announces itself. A modest mouth in the rock, leading into chambers that go back, perhaps, 40 meters, with a natural skylight in the ceiling of the main chamber that lets in a column of light during the day and would have made the cave warmer and more livable than a fully enclosed space. For tens of thousands of years, that combination, shelter, light, water nearby, a position above the valley floor that offered visibility and protection, made it exactly the kind of place that humans seek out when they need somewhere to rest. And so they came. Different kinds of humans, at different times, across a span of time so long that the words different times do not quite capture it. Russian archaeologists began working at Denisova Cave in the 1980s. Under the direction of Anatoly Derevyanko of the Institute of Archaeology and Ethnography in Novosibirsk. What they found, over decades of careful excavation, was a stratigraphy, a layered sequence of sediments that read like a compressed archive of human presence in this part of Asia, layer after layer, each one older than the one above it, each one containing the residue of the people who had sheltered here: stone tools, animal bonds, charcoal from ancient fires, and, occasionally, the bones of the people themselves. The sediments in Denisova Cave span, by current estimates, at least 300,000 years of intermittent occupation, 300,000 years of different groups returning to the same sheltered space above the same cold river. That number is almost impossible to hold in the mind. To put it in some kind of proportion, 300,000 years ago, Homo sapiens were only beginning to emerge in Africa. The fossil evidence from Jebel Irhoud in Morocco, dated to approximately 300,000 years before the present, represents some of the earliest known traces of our species anywhere on Earth. We were new to the world, barely present, still finding our shape. What had already been present in Asia, the Timon of Denis, is the wide. For a very long time before that, were other kinds of humans, other kinds of minds, other ways of moving through the world and making sense of it. And some of them came to this cave. In 2010, a paper was published in the journal Nature that changed, in a fundamental way, what scientists understood about human prehistory in Asia. The paper, led by Svante Pääbo of the Max Planck Institute for Evolutionary Anthropology in Leipzig, announced the discovery of a new human lineage, identified not from a skull, not from a skeleton, but from a fragment of bone so small it had almost been overlooked, a piece of a finger, the distal phalanx of a child's little finger, recovered from a layer of sediment in Denisova cave that was at least 50,000 years old. From that fragment, Pääbo and his team extracted ancient DNA and sequenced it. And what they found was that this child did not belong to any known human lineage. The genome was distinct from Neanderthals, distinct from Homo sapiens. It was something else, something that had been living in Asia for a very long time without anyone in the modern world knowing it existed. They called this lineage the Denisovans. After the cave where the evidence was found, a child's finger bone. That is what it took to rewrite the human story in Asia. Svante Pääbo would go on to receive the Nobel Prize in Physiology or Medicine in 2022, specifically for his discoveries concerning the genomes of extinct human lineages and human evolution. It was a recognition of work that had taken decades and that had, in a very real sense, changed what we understand ourselves to be. But the discovery of the Denisovans was only the beginning of what Denisova Cave would reveal. Because the same cave, in different layers of sediment, across different periods of time, also contained evidence of Neanderthals, the robust, cold-adapted human lineage that had ranged across Europe and Western Asia for hundreds of thousands of years had also reached this far east, into the Altai, into this same cave, above this same river, and in layers above both, the tools and eventually the remains of Homo sapiens, our own direct ancestors, arriving later, entering a landscape that was already known, already occupied, already shaped by other kinds of human minds. 3 human lineages, 1 cave system, 1 continuous sequence of sediments stretching back across an almost incomprehensible depth of time. It is worth being precise about what that means and what it does not mean. The 3 lineages did not necessarily shelter in this cave at the same time. The layers that contain their evidence are separated, in some cases, by thousands of years. They were not sitting around the same fire on the same night. But they came to the same place. They sheltered in the same chambers. They looked out from the same opening at the same valley below. And in some periods, the distance between them in time was not the vast gulf that separates us from the ancient world, but something smaller, something that a few generations of movement through the same landscape could have bridged. The evidence for how close they came is preserved in a single individual. In 2018, a team of researchers led by Viviane Slan and Svante Pääbo at the Max Planck Institute published findings in Nature that stopped the scientific community in a way that even the original Denisovan discovery had not quite managed. They had been analyzing bone fragments from Denisova Cave, fragments that had been catalogued and stored, waiting for the right analytical techniques to be developed. One of those fragments came from an individual they called Denny in the scientific literature. A girl. Based on the analysis, she had lived at least 50,000 years ago, possibly longer. She had survived long enough to leave a fragment of her bone in the sediments of the cave. And when the researchers sequenced her genome, they found something that required a moment of genuine recalibration. Her mother was a Neanderthal. Her father was a Denisovan. She was the direct offspring of two different human lineages, not a descendant of ancient interbreeding from thousands of years before her birth. Not someone who carried a small percentage of ancestry from another group across many generations of dilution. The literal first-generation child of a Neanderthal mother and a Denisovan father, two people from two distinct human lineages who had met in the way that people meet, and whose meeting had produced a child who was carried to term and lived long enough to leave her trace in the sediments of a cave that would hold it for 50,000 years. Denny is not a theory. She is not a model or a reconstruction. She is a genome, a specific individual whose specific biology has been read from the molecules she left in the stone. And what her existence tells us is that the boundary between these human lineages was permeable in the most fundamental way possible. That when Neanderthals and Denisovans were close enough to encounter each other, sometimes what resulted was a child. A real child, with a mother and a father who came from different worlds, and who left her bone in the dark of a cave that would not give it up for 50,000 years. The cave does not tell us how her parents met. It tells us only that she existed, that her parents existed, that the boundary we draw so clearly in our scientific categories between one human lineage and another was, in the lived experience of the people on both sides of it, something they could and did cross. Denisova Cave is still being excavated. Every field season adds to what the sediments have been holding for tens of thousands of years. What it has already given us is enough to change how we understand the human story in Asia. Not as the story of one lineage moving through empty territory, but as something more crowded, more complicated, more alive than that. A story in which, for a span of time measured in thousands of years, more than one kind of human mind was present on this continent, looking at the same mountains, breathing the same cold air, leaving their traces in the same stone, and then, un by un, falling silent. Chapter 2. Who were the Denisovans? There is a particular kind of frustration that belongs to knowing someone exists without knowing what they looked like. You have their letters, but not their face. You have the record of what they did, but not the sound of their voice. You know, they were real. You know they mattered. And yet the most basic thing, the thing that would make them feel present rather than abstract, the simple human fact of their appearance, is gone. That is the situation we are in with the Denisovans. We have their genome. We have, in extraordinary detail, the molecular blueprint of who they were at the deepest biological level. We know things about their DNA that we do not know about most people who lived 1,000 years ago, let alone 50,000. And yet we cannot tell you with confidence what a Denisovan looked like standing in front of you. We cannot describe the shape of their face, the color of their eyes, the build of their body, in the way that we can describe a Neanderthal from the dozens of skulls and skeletons that European caves have preserved. This is what makes the Denisovans unlike any other human lineage we have ever encountered. Every other group in human prehistory was discovered the way prehistory has always worked. Someone found a bone. Someone found a skull. The physical shape of the ancient person was the beginning of the story. And everything else came after. With the Denisovans, it worked in reverse. The genome came first. In 2010, when Svante Pääbo and his team published the first Denisovan genome from that fragment of a child's finger bone, what they had was a genetic sequence and almost nothing else. No skull. No yav. No limb bones that could tell them how tall these people were or how they were built. Just the molecular record of a genome that was clearly distinct from both Neanderthals and Homo sapiens, belonging to a lineage that had separated from the Neanderthal line somewhere in the range of 350,000 to 400,000 years ago, though researchers continue to refine that estimate as new data becomes available. A lineage that had been living in Asia, apparently across an enormous geographic range. For a very long time. The picture has grown since 2010, but it has grown slowly, and with a kind of incompleteness that requires constant intellectual honesty about what we actually know versus what we are inferring. What we know. We know the Denisovans were genetically distinct. Their genome shows a lineage shaped by hundreds of thousands of years of evolution in Asian environments that we are still working to fully understand. They were not a regional variant of Neanderthals. They were not an early form of Homo sapiens. They were their own thing. We know they ranged across an enormous geographic area. The evidence for this comes not only from Denisova Cave in Siberia but from a site more than 2,000 miles to the southwest. On the Tibetan Plateau, at an altitude above 4,000 meters. In 2019, a paper published in Nature reported the analysis of a fossilized jawbone found in Xiahe County in Gansu Province, China. The jawbone had actually been discovered decades earlier, in 1980, by a monk in a cave called Baishia Karst Cave, and had been held in a monastery for years before researchers were able to examine it scientifically. Its age, established through uranium series dating of a carbonate crust on the bone, was at least 160,000 years, and its identity, established not through DNA, which could not be recovered from bone that old in those conditions, but through ancient protein analysis, a technique that can identify species from molecular signatures preserved in collagen, was Denisovan, 160,000 years ago, on the Tibetan Plateau, at extreme altitude. That single find changed the geographic picture of the Denisovans dramatically. They were not confined to the caves of Siberia. They were present across a range that stretched from the cold forests of the Altai to the high-altitude grasslands of the Tibetan Plateau. A span of environments so different from each other that surviving in both required either extraordinary behavioral flexibility or significant biological adaptation, or very likely both. Subsequent sedimentary DNA analysis at Baishia Karst Cave, published in Nature in 2020 and in follow-up studies, found Denisovan-related DNA in cave sediments spanning multiple time periods, including layers potentially as recent as 45,000 to 60,000 years ago. Denisovan-related populations returning to the same high-altitude environment, generation after generation, across an enormous time span. Not visitors, a presence. We know, from the genetic signals preserved in modern human populations, that Denisovan-related populations were also present somewhere along the routes into Southeast Asia and the Pacific region. The evidence for this comes from the DNA of living people, which we will return to in later chapters. The precise geography of their presence in Southeast Asia remains less clear than their presence in Siberia and Tibet, because the tropical environments of that region are far less hospitable to the preservation of ancient bone and organic material. The genetic signal in modern populations points strongly toward their presence, but the physical evidence to confirm exactly where and when remains an active area of research. What we do not know is considerable. We do not have a complete Denisovan skull. The Xiahe mandible is a jawbone, significant and important, but it does not tell us the shape of the cranium or the overall architecture of the face. Researchers have made attempts to reconstruct Denisovan appearance using a technique called DNA methylation analysis, which can predict physical traits from epigenetic patterns in ancient DNA. A study published in Cell in 2019 by David Gochman and colleagues produced a reconstruction suggesting Denisovans may have had wide faces and a robust build. It was a genuinely innovative approach. It was also, as the researchers themselves acknowledged, an inference built on inference. Every image you may have seen of a Denisovan, every reconstruction, every artist's impression, is someone's educated guess. The bones to confirm or refute those guesses have not yet been found. This is worth sitting with for a moment. A human lineage that ranged across half a continent, that survived in Siberian winters and at Tibetan altitudes, that left enough genetic signal in modern human populations to be detected and measured tens of thousands of years after their disappearance. A lineage that shaped, in ways we are still working to understand, the biology of billions of people alive today. And we do not know what they looked like. We discovered them in 2010. The implications of that gap, between the scale of their presence in Asia and the recency of our awareness of them, are not merely scientific. They are a reminder of how much of the human story has been lost, how much remains to be found, and how provisional our understanding of the past always is. There are likely other human lineages waiting in the sediments of caves we have not yet excavated, or in the genomes of living populations whose ancestry has not yet been fully analyzed. The Denisovans were unknown to us 14 years ago. The question of what else we do not know is not rhetorical. It is the most honest question we can ask. Chapter 3. The Neanderthals at the Eastern Edge. There is a woman whose bones were found in the sediments of Denisova Cave. She was not Denisovan. She was not Homo sapiens. She was Neanderthal. And she lived in the Altai Mountains of Siberia at a time when the distance between her world and the Western populations of her own lineage, the Neanderthals of France and Spain and Gibraltar, was not merely geographic. It was genetic. It was the distance of tens of thousands of years of separation, of a population that had come east long ago and had been shaped, generation by generation, by the demands of a different landscape. Her genome was sequenced and published in Nature in 2014 by a team led by Svante Pääbo. Of all the ancient human genomes recovered so far, hers is among the highest quality, sequenced to a depth that allowed researchers to read her biology in extraordinary detail. What they found when they read it was not what they expected. Her parents were closely related, half-siblings, the analysis suggested, or a parent and a child, or an uncle and a niece. The population she came from was small enough, and isolated enough, that close relatives were producing offspring together, not as a cultural choice, as far as anyone can determine, but as a demographic reality. When a population falls below a certain size and loses regular contact with neighboring groups, the pool of available mates shrinks. What follows is not immediate catastrophe. It is slow biological stress, accumulating across generations, quietly narrowing the margin between persistence and disappearance. She was already living at the edge. This is one of the most important things the Altai Neanderthal genome tells us, and it is easy to overlook because it is a story written in statistics rather than in the kind of narrative that lodges in memory. But it matters enormously for understanding what the Neanderthal world looked like when Homo sapiens arrived in Asia. The Neanderthals, who had come east, were not a thriving, expanding population. They were a remnant, a group that had separated from the main body of the Neanderthal world a very long time ago and had been living in conditions of relative isolation since. Conditions that left their mark in the genome of a woman who sheltered in a cave above a Siberian river and left her bones there for tens of thousands of years before anyone would think to look for her. Most people think of Neanderthals as European, and it is true that the richest Neanderthal record comes from the cave systems of Western Europe. But the geographic range of Neanderthal populations at their peak extended far beyond the forests and limestone cliffs of France and Spain. It extended east through the Caucasus, into the Levant, through the uplands of Central Asia, and into the Altai, to the same mountains where Denisova Cave sits above its cold river. They had traveled far from wherever their lineage began. They had adapted, across hundreds of thousands of years, to landscapes that the founders of their line would not have recognized. At Teshik-Tash in Uzbekistan, excavated in the 1930s, the remains of a Neanderthal child were found deep in Central Asia. The precise circumstances surrounding the remains have been debated for decades in the literature. and researchers continue to discuss what can and cannot be concluded from them. What is not debated is the child's presence there, thousands of miles from the caves of Western Europe, in an environment that demanded a very different set of adaptations than the temperate forests where most Neanderthal remains have been found. They were not lost. They had chosen this. Or rather, their ancestors had, generation by generation, moved further into a continent that was already occupied by another human lineage, the Denisovans, who had been there far longer. And in the Altai, something had happened that the genome of the cave woman reveals. The Altai Neanderthal population had, at some point in its past, interbred with Denisovans. The signal is there in her genome, not as a dominant ancestry component, but as a trace, a ghost of earlier contact between the two lineages that had been present in this region. Two populations that had been living in overlapping or adjacent ranges for long enough that their members had, at some point, come together in the way that Denny's own parents would come together later. Think about what that means. By the time Homo sapiens began moving into Asia, the Neanderthals and Denisovans in this region already had a shared history. They had already crossed the boundary between their lineages. They had already produced offspring. They had already absorbed something of each other into their respective populations. The world that Homo sapiens entered was not a world of two separate and competing groups meeting for the first time. It was a world with a prior history of encounter and exchange that predated our arrival by an unknown but significant span of time. We were the newcomers. They had been there long enough to have a past together. The Altai Neanderthal woman whose genome was sequenced did not survive. Her population did not survive. The demographic stress, visible in her genome, the inbreeding, the small population size, the isolation, from the larger Neanderthal world to the West, was not something that a group in that condition could absorb indefinitely. But she was there. She sheltered in the cave above the river. She lived in the Altai Mountains, with the particular knowledge of that landscape that a lifetime of dependence on a specific place produces. She knew things about the valley below and the forests above and the movements of the animals through the seasons that no one who came after her would ever know. Because the knowledge lived in her and in the people around her, and in no other form. When she was gone, that knowledge was gone with her. That is the weight the bones carry. Not only the story of a lineage in decline, but the story of everything a life contains that does not survive the life. Chapter 4. The Arrival. Picture the ocean. Not the ocean as you know it from a coastline or a map. The ocean as it would have appeared to a small group of people standing on a shore somewhere in what is now Southeast Asia, somewhere between 60,000 and 70,000 years before the present. A body of water so vast that looking at it from the shore gave no useful information about what lay beyond it. Because what lay beyond it was simply more water. Horizon after horizon, until the curvature of the earth itself hid whatever came next. And yet, someone looked at that ocean and decided to cross it. Not on a whim. Not by accident. With watercraft of some kind. With enough knowledge of the sea to survive days or weeks on open water. With enough planning to bring food and fresh water for people who could not know exactly how long the crossing would take. The people who first reached Australia, arriving on a continent that no human being had ever stood on before, were Homo sapiens. And they arrived, based on current evidence, somewhere between 50,000 and 65,000 years ago, making their crossing one of the earliest confirmed achievements of maritime navigation in the human record. They crossed. They arrived. They stayed. And the DNA of their descendants, Aboriginal Australians and Papuans living today, carries among the highest levels of Denisovan-related ancestry of any modern population on Earth. Before we follow that thread, which we will in the chapters ahead, it is worth standing at the shore for a moment longer. Because the people who looked at that ocean and decided to cross were not arriving from nowhere. They had been moving for a very long time, across landscapes that were already occupied, through territories where other human lineages had been present for far longer than our own. The movement of Homo sapiens into Asia was not a tide. It was not a campaign. It was a process that unfolded across tens of thousands of years, in different regions, at different times, driven by the same forces that have always moved populations across landscapes: the search for food, the pressure of climate, the slow, generational drift of people following resources into territory that was accessible, and pulling back from territory that was not. At Tam Pa Ling Cave in the Annamite Mountains of Laos, researchers have found fragmentary human remains that have been dated using multiple methods, including uranium series dating and luminescence dating of the surrounding sediments, to somewhere between 63,000 and 86,000 years before the present. These dates remain subject to ongoing discussion in the research literature, as dating at this range and in these conditions carries significant uncertainties. But if the evidence holds under further scrutiny, it places our species in Southeast Asia far earlier than the older models assumed, at a time when Denisovan-related populations were almost certainly still present somewhere in the region, not ahead of us, alongside us. In the islands of Southeast Asia, the picture was more complex still. Homo floresiensis, the small-bodied hominin whose remains were found in Liang Bua Cave on the Indonesian island of Flores and published in Nature in 2004, appears to have survived until at least 50,000 years ago, placing their presence squarely within the time frame of Homo sapiens' expansion through the region. Whether the two populations ever encountered each other is not established by current evidence. The record at Liang Bua shows the Homo floresiensis remains ceasing before the appearance of Homo sapiens remains. But the precision of the dating is not sufficient to rule out a period of overlap. The question remains open. On the island of Luzon in the Philippines, remains found at Calao Cave and published in Nature in 2019 represent a previously unknown human lineage designated Homo luzonensis. The remains date to approximately 67,000 years before the present. How long this lineage persisted beyond that date, and whether their presence overlapped with incoming Homo sapiens populations, has not yet been established. What emerges from these discoveries is not the picture of a single wave of modern humans sweeping through empty territory. It is a picture of a continent that was already full. Multiple distinct human presences, each adapted to specific island or regional environments, each representing a long history of evolution in a specific place. The Denisovans across a vast range from Siberia toward the Pacific. The Neanderthals from Europe deep into Central Asia. Homo floresiensis on Flores. Homo luzonensis in the Philippines. Possibly others we have not yet found. All of them present. All of them. In some regions and some periods. Present at the same time as our arriving ancestors. The genetic evidence confirms the overlap beyond reasonable doubt. The DNA that modern humans carry from Denisovan-related populations did not get there through some ancient and distant common ancestor predating the split of these lineages. It got there through actual contact, actual meetings between actual people, that produced children who grew up and had their own children, and passed the genetic contribution of those meetings forward across tens of thousands of years to the people alive today. The meetings happened. We are, in part, their consequence. Chapter 5. What happens when three lineages share a continent? Come back to Denny for a moment. Not to her genome, not to the scientific paper, not to the analytical techniques that made her visible to us across 50,000 years of darkness. Come back to her as a person. A child who was born in the Altai Mountains of Siberia, who had a mother and a father, who was held, in all likelihood, by hands that cared about whether she was warm and fed and safe. who lived long enough, in a world that was cold and difficult and demanded everything its inhabitants had, to leave a fragment of her bone in the sediments of a cave. Her mother was a Neanderthal. Her father was a Denisovan. That is the most direct evidence we have for what it actually meant. In human terms, for multiple lineages to share a continent. Not a theory about population dynamics. Not a model of competitive exclusion. A girl, with a mother and a father who came from different worlds, who met somewhere in the cold landscape of the Altai, and whose meeting produced a child who carried both of them in her cells. We do not know how her parents met. We do not know whether their encounter was a single crossing of paths or something longer. We do not know what language or gesture passed between them, or whether the concept of difference was something either of them carried in any form we would recognize. What we know is that they met, and that what came from that meeting was a daughter who was born into the world and lived and left something behind. The analysis of Denny's genome revealed something further that deepens the story. Her Denisovan father himself carried a trace of Neanderthal ancestry, suggesting that the population he came from had experienced some degree of interbreeding with Neanderthals in an earlier period. The boundaries were not only crossed in the generation that produced Denny. They had been crossed before, in different places and at different times, by different individuals in different circumstances, with results that propagated forward into subsequent generations. Denny was not the beginning of the story between these lineages. She was one moment in a longer story that had been unfolding for longer than we can trace precisely. The genetic record in living people extends this picture far beyond a single cave in Siberia. People who trace their ancestry to populations that passed through or settled in Asia carry DNA from Denisovan-related populations. The distribution of that ancestry across modern populations is not random. It is geographically structured in ways that tell us something about where the contact occurred and roughly when. Populations from Melanesia, Papua New Guinea, and Aboriginal Australia carry among the highest levels of Denisovan-related ancestry of any groups analyzed so far, with many studies estimating somewhere in the range of 3 to 5%. Though the precise figures depend on the reference genome and methodology used and should be understood as approximations. Populations from mainland East Asia carry lower but detectable amounts. South Asian populations carry intermediate levels. The gradient itself is a piece of evidence. It points toward substantial Denisovan-related populations in the regions that the ancestors of Melanesians and Australians passed through. Populations present enough that contact occurred at a scale that left a clear signal tens of thousands of years later. A study published in Cell in 2018 by Sharon Browning and colleagues at the University of Washington found evidence consistent with at least 2 distinct episodes of Denisovan-related ancestry entering modern human populations, one contributing more to populations from Papua New Guinea and Australia, another contributing to populations from mainland East Asia. If this interpretation holds under further scrutiny, the contact between our ancestors and Denisovan-related populations was not a single event in a single place. It was a pattern, recurring across different regions, across different generations, across a span of time and geography that we are still working to map precisely. Now, it is important to be honest about what the evidence does and does not tell us about the nature of those encounters. The genetic record tells us the outcome. It does not tell us the character. We cannot know whether the meetings between Homo sapiens and Denisovan-related populations were frequent or rare, whether they happened across a broad range or only in specific places where the populations came into close proximity, whether they were understood by the people involved as encounters with another kind of human being or experienced as something else entirely. We do not know what it felt like to be on either side of those boundaries. What we do know is that the outcome of those encounters was not neutral. Some of what crossed the boundary between the populations was retained. Some of it was retained at frequencies higher than chance would predict, which is the signature of natural selection, of variants that were useful to the people who carried them. The Denisovans and Neanderthals had been in Asia for hundreds of thousands of years before Homo sapiens arrived. They had learned, in the only way biological populations learn, through the slow and unintentional selection of variation across many generations, how to survive in specific Asian environments. That learning was encoded in their genomes, and portions of their genomes passed, through the encounters we have been describing, into the populations that came after them. The people who came after were helped, in ways they could not have known, by what the people before them had carried. We will spend the next chapters looking at exactly what was transferred and what it has meant for the billions of people alive today who carry that inheritance. But before we do, sit for a moment with the larger picture. 3 human lineages, sharing a continent, not in harmony, not in warfare, not in any simple relationship that our categories fully capture. In proximity, in the complicated, unplanned, generationally extended way that populations share landscapes when they are both present in the same place across the same span of time. Denny's parents were part of that proximity. Their daughter was its product. And in the genomes of billions of people alive today, the record of everything that was transferred across those encounters. Everything that survived, the distance between their time and ours, is still present, still readable, still, in the most literal biological sense, alive. Chapter 6. The gift hidden in altitude. There is a plateau in Central Asia that sits so high above the rest of the world that the air itself becomes a problem. The Tibetan Plateau covers an area roughly the size of Western Europe at an average elevation of more than 4,000 meters above sea level. At that altitude, the atmosphere contains approximately 40% less oxygen than it does at sea level. For most human beings, prolonged exposure to those conditions produces a cascade of physiological responses that range from unpleasant to life-threatening. Headaches, fatigue, disrupted sleep. In severe cases, fluid accumulates in the lungs and brain. The body, built for the oxygen-rich air of lower elevations, struggles to extract enough of what it needs from air that does not provide enough of it. And yet, for thousands of years, people have lived on the Tibetan Plateau, not as visitors passing through, as permanent residents, raising families, herding animals, building communities at elevations that would incapacitate most of the rest of humanity within days. The question of how they do it has occupied biologists and physiologists for decades. Part of the answer pointed somewhere unexpected. It pointed to Denisovan-related ancestry. In 2010, a team of researchers led by Rasmus Nielsen at the University of California, Berkeley, published a study in Science identifying a gene variant that appeared to be responsible for the remarkable altitude adaptation of Tibetan people. The gene is called EPAS1, and it encodes a protein that plays a central role in the body's response to low oxygen conditions. The version of EPAS1 found in most humans causes the body to respond to low oxygen by producing more red blood cells, thickening the blood in ways that can, over time, cause cardiovascular problems at sustained altitude. The version found in Tibetans does something different. It allows the body to use oxygen more efficiently without the problematic overproduction of red blood cells, enabling sustained life at high altitude without the physiological costs that high-altitude living imposes on people who carry the standard variant. What was striking about this Tibetan variant was not only what it did, but how common it was. In the Tibetan population studied, approximately 87% of individuals carried it. In lowland Han Chinese, the closest neighboring population with whom Tibetans share much of their recent ancestry, the figure was around 9%. That gap, between 87% and 9%, is not the kind of difference that accumulates through ordinary random variation across a few thousand years of separation. It is the signature of strong natural selection. of a variant that conferred such a significant survival advantage in a specific environment that the individuals who carried it survived and reproduced at higher rates than those who did not. Generation after generation, until it became nearly universal in the population living in that environment. The variant was doing something important. The question was where it had come from. In 2014, a study published in Nature by a team that included Emilia Huerta Sanchez and Rasmus Nielsen at UC Berkeley compared the Tibetan EPAS1 variant against the genomes of other human populations, including the ancient genomes sequenced from Denisova cave. What they found was that the Tibetan variant was not closely related to any version of EPAS1 found in other modern human populations. It was, however, closely related to the version found in the Denisovan genome. The most likely explanation, the researchers concluded, was that this variant had entered the ancestral Tibetan population through contact with a Denisovan-related lineage, that at some point in the history of the people who would become the Tibetan population, their ancestors had encountered individuals from a Denisovan-related group, and through that encounter had acquired a version of EPAS1 that the Denisovan lineage had been carrying. And this variant, once present in the Homo sapiens population moving into high-altitude environments, was powerfully selected for because of the advantage it conferred at extreme elevation. Now, it is important to be precise about what this finding does and does not establish. It establishes that the EPAS1 variant in Tibetans is of Denisovan-like ancestry. That is supported by the genomic evidence and is held up under subsequent scrutiny. It does not establish with certainty that Denisovan-related populations were themselves physiologically adapted to high altitude in exactly the way that modern Tibetans are. The variant may have been present in Denisovan populations without being under strong selection in their particular circumstances, carried as a neutral or mildly beneficial variant that only became powerfully advantageous when Homo sapiens populations moved into the extreme altitudes of the Tibetan plateau. The Shahe mandible, found at an altitude above 3,000 meters and dated to at least 160,000 years before the present, suggests that Denisovan-related populations were present on or near the Tibetan plateau for a very long time. But the precise relationship between their biology and high-altitude adaptation remains an area of active research rather than settled conclusion. What can be said is this: a variant of a gene that is now found in approximately 87% of Tibetans. A variant that allows those people to live and thrive at altitudes that would compromise the health of most human beings, appears to have entered the Tibetan gene pool through contact with a Denisovan-related lineage. That lineage had been present in the region for at least 160,000 years before that contact occurred. The variant they carried, once transferred into the Homo sapiens lineage, conferred an advantage so significant that natural selection amplified it until it became nearly universal in the population that needed it most. Think about the distance that represents, not geographic distance, though that is real, temporal distance. The Shahe individual lived on the Tibetan plateau at least 160,000 years ago. The modern Tibetan people who carry the EPAS1 variant are alive today. The chain of biological transmission that connects them crosses a gap of time longer than the entire span of anatomically modern human presence in many parts of the world. Something that was present in a Denisovan-related body on the Tibetan plateau more than 100,000 years ago is present in Tibetan bodies today. Not as a memory, not as a cultural inheritance, as a functioning piece of molecular machinery, doing its work in the cells of people who have never heard the name Denisovan and who learned about the existence of this ancient lineage, if they learned about it at all, from the same recent scientific literature that the rest of the world learned it from. There are approximately 7 million people living on the Tibetan plateau today. A significant majority of them carry this variant. Each of them is, in this specific and measurable biological sense, a continuation of something that began in a human lineage that no longer exists as a distinct population in the world. The gift was given without knowing it was a gift. It was received without knowing it had been given. And it has been working, quietly and continuously, for longer than most of human civilization has existed. Chapter 7. The Immune Memory of Asia. If you trace your ancestry to Southeast Asia, to the islands of Melanesia, to Papua New Guinea, to Aboriginal Australia, or to any of the populations that moved through those regions in the deep past, then, somewhere in your genome, woven into the molecular instructions that define your biology, there are sequences that were shaped by hundreds of thousands of years of Denisovan-related evolution, not a trace amount, not a rounding error in the genetic record, a measurable, documented contribution from a human lineage that most of the world did not know existed until 2010. The geographic pattern of Denisovan-related ancestry in modern human populations is one of the most informative pieces of evidence we have about where Denisovan-related populations actually were and how extensive their presence in Asia may have been. And that pattern, when you look at it carefully, tells a story that the fossil record alone could never tell. Populations from Melanesia and Papua New Guinea carry among the highest levels of Denisovan-related ancestry of any groups analyzed so far, with many studies estimating somewhere in the range of 3 to 5%. Though the precise figures depend on the reference genome and methodology used and should be understood as approximations rather than fixed numbers. Aboriginal Australians carry comparable levels. Populations from mainland East Asia carry lower but detectable amounts. South Asian populations carry intermediate levels. The gradient from high in Melanesia and Australia to lower in mainland East Asia is itself a piece of evidence about the geography of ancient contact. The simplest interpretation, and the one that the current research literature most broadly supports, is that Denisovan-related populations were present somewhere across a wide range in Asia, with substantial populations somewhere along the routes into Southeast Asia and the Pacific. As Homo sapiens populations moved through those regions, contact occurred at a scale that left a clear genetic signal tens of thousands of years later. Now, the fact that Denisovan-related DNA is present in these populations is established. The more interesting question, from the perspective of what that ancestry actually means for the people who carry it, is what those sequences do. Because not all ancient DNA that enters a population persists at high frequency, much of it is carried along by chance across generations without conferring particular advantage or disadvantage, but some of it is retained at frequencies higher than chance would predict, which is the signature of natural selection, of variants that were useful to the people in whose bodies they were working. Research published in PLOS Genetics and in subsequent studies has identified Denisovan-related variants in genes associated with immune function, specifically in regions involved in the innate immune system, the body's first-line response to pathogens, at frequencies in certain populations consistent with those variants having been retained because they were beneficial. Analysis of the toll-like receptor gene family, which plays a fundamental role in detecting and responding to bacterial and viral threats, has found Denisovan-related contributions in non-African populations at frequencies that some researchers interpret as evidence of positive selection. The hypothesis that connects these findings is straightforward in its logic, even if the full causal chain has not been confirmed in all its details. Denisovan-related populations may have carried immune variants shaped by long exposure to Asian environments. When Homo sapiens arrived in those same regions and contact occurred between the populations, some of the Homo sapiens individuals who descended from those encounters inherited those variants. In the disease environments of tropical Asia, variants shaped by a much longer prior exposure may have helped their carriers respond more effectively to pathogens that the incoming Homo sapiens populations had not previously encountered. The individuals who carried those variants may have survived at higher rates. Their descendants carried the variants forward. Over thousands of generations, the variants that were most beneficial in specific environments were retained at frequencies above what random chance would predict. It is important to be honest about the limits of what we can say here. The relationship between specific Denisovan-related variants and specific immune functions in modern populations is an active area of research, and the precise causal connections are complex and not fully resolved. The presence of a variant at elevated frequency is consistent with positive selection, but does not by itself tell us exactly what selective pressure produced it or what specific survival advantage it conferred in the ancestral environment. The research literature is careful about these distinctions, and this account should be as well. What can be said is that the pattern is there, and the most plausible explanation for why those particular variants were retained connects directly to the long presence of Denisovan-related populations in the environments where the people who carry them were living. Think about what that means in practical terms. Denisovan-related populations may have spent hundreds of thousands of years somewhere across the routes into Asia and the Pacific. During that time, they would have been exposed to disease environments that were, in many ways, biologically demanding, in ways that colder environments further north were not. The immune variants they may have carried, shaped by that long prior exposure, passed in part into the populations that came after them, outlasted them, and absorbed them. The people who came after were, in certain specific and measurable ways, better equipped to survive in those environments because of what the populations before them had carried. There is something in that which resists easy categorization. It is not quite tragic. Because the benefit was real, people survived challenges they might otherwise not have survived. Populations persisted in environments that might otherwise have been more costly to inhabit. But it is not quite redemptive either, because the populations whose biological inheritance was transferred did not themselves survive as distinct lineages. The gift was transferred. The givers were not. What persists is not them. It is something of what they may have carried across hundreds of thousands of years, preserved in molecular form, passed forward into a future that belonged to someone else. The people of Melanesia and Papua New Guinea and Aboriginal Australia are not Denisovans. They are fully themselves, with all the history and culture and identity that entails. But they carry, woven into their biology, a contribution from human populations that lived in their part of the world for an almost incomprehensible length of time before their ancestors arrived. That contribution is simply there, in their cells, doing the molecular work that it has been doing for tens of thousands of years, quietly, continuously, long after the populations who provided it fell silent. Chapter 8. The Silence After. There is a particular kind of ending that leaves no monument. Not the ending of a battle, which leaves bones and weapons in the earth. Not the ending of a city, which leaves walls and ash and the outlines of streets that archaeologists can trace centuries later. Not even the ending of a language, which leaves at least the shapes of words in the mouths of the last people who spoke it before it was gone. The ending we are talking about tonight is quieter than any of those. It is the ending of a population, the slow, generational dimming of a group of human beings who became fewer and then fewer still, until the number that remained was not enough to sustain what had existed. And then there was nothing left to sustain. We know this ending happened to the Denisovan-related populations of Asia. We know it happened to the Neanderthals. We know it happened to Homo floresiensis on Flores and to Homo luzonensis in the Philippines and to whatever other human lineages were present in Asia whose traces we have not yet found. What we do not always acknowledge is how little we know about how it happened, and how different the endings of these different lineages appear to have been from each other. The Neanderthal disappearance in Europe has a rough chronology. The sites that contain evidence of their presence stop producing that evidence at roughly 40,000 years before the present. The physical remains of Neanderthal individuals become absent from the layers that continue to accumulate in the caves they had sheltered in for hundreds of thousands of years. It was not an instant. It was a long contraction. A population pulling back toward its final refuges in the south and west of Europe, becoming fewer and more isolated, until the isolation itself became the final condition. The Denisovan disappearance is different. It is different because we do not have a comparable archaeological record to watch go silent. The sedimentary DNA evidence from Baishia Karst Cave on the Tibetan Plateau suggests Denisovan-related populations were present there until perhaps 45,000 to 60,000 years ago. Beyond that, the record is sparse. Not because nothing is there, because we have not yet developed the tools and conducted the fieldwork necessary to find it. This distinction matters enormously. The tropical and subtropical environments of Southeast Asia, where the genetic evidence in modern populations points most strongly toward a substantial Denisovan-related presence, are profoundly hostile to the preservation of ancient bone and organic material. Heat and humidity and biological activity decompose organic matter rapidly. The caves of tropical Southeast Asia have not been subjected to the same intensity of systematic archaeological investigation as the cave systems of Europe. The sedimentary DNA techniques that have proven so powerful at Denisova Cave and Baishia are only beginning to be applied to sites in this region. The date at which the last Denisovan-related populations ceased to exist is genuinely unknown. Not imprecisely known. Unknown in a deeper sense. Because the data to answer the question has not yet been fully collected. What we can say is that by the time any kind of historical record becomes available for the populations of Southeast Asia and the Pacific, there are no Denisovans. The lineage that had been present in Asia for hundreds of thousands of years is absent, and the populations who came after, who absorbed them or outlasted them, carry Denisovan-related ancestry in their genomes as the only direct record of what had existed in those landscapes before. The situation with Homo floresiensis adds another dimension to the story of endings in Asia. The remains found in Liangbua Cave appear to place the final occupation of that site at roughly 50,000 years before the present, well within the time frame of Homo sapiens' expansion through the region. Whether Homo sapiens and Homo floresiensis ever encountered each other is not established. The record shows the Homo floresiensis remains ceasing before the appearance of Homo sapiens remains. But the precision of the dating is not sufficient to rule out a period of overlap. The question remains open. What the existence of these different lineages illustrates is that Asia was not a continent with one or two competing human populations at the time of our arrival. It was a continent of multiple distinct human presences, each adapted to specific environments, each representing a long history of evolution in a specific place. All of them are now gone as distinct populations. The question of what ended them involves several contributing factors that the current evidence cannot weigh precisely against each other. Climate change was certainly a factor across this period. The time between 50,000 and 30,000 years ago included significant climatic shifts that altered the environments these populations depended on. Populations already small and fragmented would have been particularly vulnerable to deterioration in the resources available to them. The arrival of Homo sapiens populations, with their larger networks and potentially more flexible strategies, introduced a form of competitive pressure that, even without direct conflict, could have tipped the demographic balance for populations already living close to their limits. Disease almost certainly played a role, though the evidence for this is indirect and the magnitude of its contribution is genuinely unknown. Contact between populations separated for long periods brings pathogens into contact as well. The precedents from much later periods of human history suggest this factor should not be dismissed, even when direct evidence is absent. And then there is the factor that is most difficult to quantify and most important to hold honestly. We do not know what we do not know. The Denisovans were unknown to science until 2010. Homo luzonensis was unknown until 2019. Each discovery has expanded the picture in ways that could not have been anticipated. The gaps in our current understanding are invisible from where we stand, which means we cannot know how large they are. What we can know is that the silence after is real, and that it is not the silence of populations that left nothing behind. It is the silence of populations who left what they had in the only form that could cross the distance between their time and ours. Not in stone, not in story, in the genome, in the cells of people alive today, on the Tibetan Plateau and in the islands of Melanesia and in the highlands of Papua New Guinea, where something of what those ancient populations carried is still present and still working. Tens of thousands of years after the last of them drew their last breath in a world that has changed almost beyond recognition since. Chapter 9. What the Cave Still Holds. In 1980, a monk found a jawbone in a cave on the Tibetan Plateau. He did not know what it was. He brought it to a monastery, where it was kept for decades, unremarkable among other objects. Waiting. It was not until 2019 that researchers were able to examine it with the tools that would reveal what it actually was. 160,000 years old. Denisovan. Evidence of a human presence on the roof of the world that no one had suspected existed. The jawbone had been there the whole time. That is the nature of what the caves still hold. Not mystery in the dramatic sense. Something quieter than that. Objects and sediments and molecular traces that have been patient in a way that only geology can be patient. Waiting not for the right person, but for the right tools. The right questions. The right moment in the long development of human science, when what they contain can finally be read. Denisova Cave is still being excavated. Russian and international research teams continue to work through its sediments, moving carefully through material that cannot be replaced if it is damaged or destroyed by hasty excavation. Each field season adds something. Each layer opened reveals the residue of people who sheltered there across an almost incomprehensible span of time. And the tools available to read what the cave holds are changing faster than at any previous point in the history of the discipline. Consider what sedimentary ancient DNA has made possible. Before this technique matured, finding evidence of an ancient human population required finding their bones. Bones are rare. They preserve poorly in many environments. They are found by chance as much as by design. The entire Denisovan lineage was identified from a handful of bone fragments, any one of which might easily have been missed or discarded in an earlier era of archaeology. Sedimentary ancient DNA changes that equation. Ancient DNA shed from skin, hair, and other organic material accumulates in cave sediments and can, under the right preservation conditions, survive for tens of thousands of years. By extracting and sequencing DNA from carefully collected sediment samples, researchers can detect the presence of Denisovan-related populations in a layer of cave floor even when no physical remains of those individuals have been found. The results at Baishia Karst Cave, on the Tibetan Plateau, illustrate what this means in practice. Researchers found Denisovan-related DNA in sediment layers spanning from over 100,000 years ago to potentially as recent as 45,000 years before the present. Not from bones, from the molecular traces left behind in the dirt of a cave floor. Preserved across a span of time longer than all of recorded human history. Denisovan-related populations on the Tibetan Plateau, across an enormous time span, returning, generation after generation, to the same high-altitude cave, above the same cold landscape. That record was invisible until the tools to read it were developed. Researchers are now applying sedimentary DNA analysis to cave sites across Southeast Asia, where the genetic evidence in modern human populations most strongly suggests that substantial Denisovan-related populations once existed. The caves of Sumatra, Borneo, and the Philippine archipelago are being examined with tools that did not exist a decade ago. Whether those sediments have preserved the molecular traces of Denisovan-related populations across tens of thousands of years of tropical conditions is a question the research is only beginning to approach. Ancient protein analysis. The same technique used to identify the Xie mandible when ancient DNA could not be recovered is being applied more broadly to existing fossil collections across Asia. Fossils sitting in museum drawers for decades, catalogued under older taxonomic categories that predate our knowledge of the Denisovans, may carry protein signatures that would identify them differently if examined with the right tools. The possibility that Denisovan-related remains are already in museum collections, unrecognized because they were found before the tools to recognize them existed, is one that researchers take seriously. Think about what that means. Somewhere in a storage room, in a museum, in a drawer that has not been opened in years, there may be a bone that was collected in the 1950s or the 1970s by a researcher who had no way of knowing what it was. A bone that has been sitting in darkness ever since. Patient in the way that only stone and bone can be patient. Waiting for someone to come looking with the right question. That bone may be Denisovan. We may already have it. We just do not know it yet. The cave at Denisova itself has yielded, in less than 50 years of systematic investigation, a new human lineage. The first confirmation of first-generation interbreeding between two distinct human lineages, a genome that revealed the genetic contributions of an extinct population to billions of living people, and a window into a period of human history that was, until very recently, almost entirely invisible. And researchers estimate that only a fraction of the cave's total sediment volume has been excavated. The cave is patient. It has been accumulating its record for 300,000 years. It will still be there tomorrow, and the year after, and for as long as it takes to develop the tools and ask the questions that will allow the rest of what it holds to speak. The past does not disappear. It accumulates, and we are, slowly and with increasing precision, learning to read what it has been keeping for us. Chapter 10. What we are made of. Come back to Denny one last time. Not to the scientific paper. Not to the genome sequence. Not to the analytical framework that made her visible to us across 50,000 years. Come back to her as a child who was born, who had a mother who was Neanderthal and a father who was Denisovan, and who came into the world in the Altai Mountains of Siberia in a time before writing, before agriculture, before any of the things we associate with the development of civilization, who was held by hands that cared whether she was warm, who grew up in a world that was cold and demanding and required everything its inhabitants had simply to survive another season, who left a fragment of her bone in the sediments of a cave, and who waited, in the patient way that only the dead can wait, for 50,000 years, until a team of researchers extracted her genome from that fragment and read the story it contained. Her existence tells us something that no amount of population modeling or statistical analysis can tell us as directly. It tells us that the boundaries between these human lineages were not walls. They were membranes, permeable in both directions, crossable by ordinary people in ordinary circumstances, in the way that people have always crossed the boundaries between the groups they were born into when the circumstances brought them close enough. Denny's parents crossed that boundary, and the record of their crossing is in the genomes of billions of people alive today. If your ancestry traces to populations that passed through Asia, somewhere in your genome there are sequences shaped by hundreds of thousands of years of Denisovan-related evolution. The EP has one variant that allows Tibetan people to live at extreme altitude, present in approximately 87% of the Tibetan population, traceable through genomic analysis to a Denisovan-related origin. The immune variants in populations from Melanesia and Southeast Asia retained at frequencies consistent with positive selection, pointing toward a long prior Denisovan-related presence in those disease environments. The toll-like receptor variants found across non-African populations, carrying contributions from both Neanderthal and Denisovan-related sources, doing their molecular work in immune systems across the world. These are not relics. They are not museum pieces preserved behind glass. They are functioning components of living biology, doing their work right now, in the bodies of people who are alive tonight, on the Tibetan Plateau, and in the islands of Melanesia, and across the vast and varied landscape of a continent that once held more kinds of human beings than we knew existed until very recently. Now consider what that means for the story we tell about ourselves. The version of that story most of us absorbed, somewhere between childhood and adulthood, went something like this. Homo sapiens left Africa. They were more adaptable, more creative, more capable than the other human lineages they encountered. They spread across the world, and the other lineages disappeared. And one kind of human mind inherited the earth. The genetic evidence tells a different story. Not a story of replacement. A story of absorption, of exchange, of populations that met across the enormous geography and extended timescale of the human presence in Asia, and that passed something across the boundaries between them. And that continued, carrying the evidence of those meetings in their cells across tens of thousands of years to the present moment. At least for Homo sapiens whose ancestry passes outside of Africa, the history of our species has never been the history of a single, poor, unbroken line. It has been the history of encounter, of biological boundaries that were real but permeable, of people who met, in the various ways that people meet, and pass something forward into a future they would not live to see. The Neanderthals contributed. The Denisovans contributed. And quite possibly other lineages whose existence we have not yet confirmed contributed as well, in ways that are still present in the genomes of living people and that we have not yet learned to read. We are the product of all of that. Not only of the Homo sapiens lineage that left Africa and spread across the world, but of the populations who were already here when our ancestors arrived, who had been here, in some cases, for hundreds of thousands of years, who had learned, in the only way biological populations learn, how to survive in the specific environments of the specific places where they lived, and who passed that learning forward, in the only form that could survive the distance between their time and ours. The cave at Denisova is still there. The sediments are still accumulating, layer by layer, as they have been for 300,000 years. The researchers who work there are still working. Every season, they go deeper into the record. Every season, something new becomes visible that was not visible before. And in laboratories around the world, in the sequences of genomes extracted from ancient bones and from the blood of living people, the record of what happened in Asia tens of thousands of years ago is still being read. We are not at the end of this story. We are somewhere in the middle of understanding it. And the part we have understood so far, the jawbone in the monk's monastery, the finger bone of a child in a cave in Siberia, the genome of a girl whose parents came from different worlds, the altitude adaptation of 7 million people on the Tibetan Plateau, the immune memory carried in the cells of people across the Pacific. All of it is already enough to change, in a fundamental way, how we answer the question that underlies all of human prehistory, the question of who we are. The answer is not simple. It was never simple. We are the people who crossed an ocean to reach Australia before anyone else had found it. We are the people who looked at the Tibetan Plateau and stayed. We are the people who moved through the forests and the island chains of Southeast Asia and encountered, in the populations already present there, something that passed into us and has not left. We are Denny's world. Continued. Not by replacement, not by conquest, by the slow, unplanned, generationally extended process of populations in proximity, passing something forward across boundaries that were real but never. In the end, absolute. The encounters happened. The boundaries were crossed. Something passed through them and survived. And it is in you, right now. As you listen to this, in whatever hour surrounds you, in whatever dark or half-light you have settled into for the night. Not as memory. Not as feeling. Not as anything you can consciously access or deliberately recall. As biology. As the molecular inheritance of meetings that occurred before writing existed. Before agriculture existed. Before any of the things we associate with civilization had been invented by anyone anywhere on Earth. As something older than all of that. And more durable. Let that be the last thought you carry as you drift off tonight. Not the vastness of the time involved, which is too large to feel properly. Not the complexity of the science, which is real but not what matters most right now. Just the simple, quiet, extraordinary fact. They were here. For hundreds of thousands of years. In different forms and in different places across this vast continent. They were here. And something of what they were is still here. In you. Sleep well. Good night.